Hello, everyone, and welcome to the second in our series of podcasts where we delve into some of the interesting pathways in cancer biology and look at the emerging clinical data to see where the space is going. I'd like to introduce our Chief Medical Officer here at Insilico, Sujata Rai. Sujata, would you like to introduce yourself?
Yes, hi. I'm the head of global clinical development here at Insilico, Chief Medical Officer. Thanks, Sean.
And I'm Sean Cullen, Associate Director of BD here at Insilico. So the goal of this session really is to introduce the HIPPO pathway, HIPPO signaling pathway in cancer biology, and how this promising area is being targeted in the drug development space. And with that, I will pull up the deck.
So, Sean, I'm a little intrigued about the unusual name for the pathway. How did that come about?
Yeah, sure, Sujata. So the name certainly is a little quirky. So this comes from some early academic research done on some of the pathway components. So this pathway was discovered originally in fruit flies, Drosophila melanogaster, in the 1990s through genetic screening for genes that regulate organ development and tumor suppression. The Hippo signaling pathway coordinates cell proliferation and regeneration and both cellular and immune homeostasis. And because of these important functions, it's also a central regulator of organ size. Now, the pathway actually takes its name directly from one of its key signaling components, the protein kinase HIPPO or HPO. And mutations in this gene were found to lead to tissue overgrowth or hippopotamus-like phenotype in the fruit flies, as depicted on the left-hand side graphic there. While this seminal work was done in fruit flies, orthologs of the hippo pathway components have subsequently been discovered in mammals.
Very interesting, Sean. I guess it does look like a hippo with the overgrowth of tissue and the hard-looking outer skin.
Yeah, sure. A lot of genes discovered in fruit flies earlier on were given names that describe the shape that gene mutations conferred in the flies. So some of these names tended to be quite imaginative. But as I mentioned earlier, Sujata, orthologs of these Drosophila genes were subsequently discovered to play a role in human cancer. Can you tell us about some of the human cancers where HIPPO pathway dysregulation plays a role?
So we see dysregulation of the HIPPO pathway across a number of solid tumors where TEAD inhibitors may be effective as monotherapy. So for example, in malignant mesothelioma, NF2 mutations are seen in approximately 40% of patients. Similarly, YAP-TAZ fusions are seen in over 90% of patients with epithelioid hemangioendothelioma. We also see other mutations associated with Hippo pathway dysregulation in head and neck squamous cell cancer, endometrial cancer, and non-small cell lung cancer. We also know that there is preclinical data that demonstrates upregulation of TEAD activity after targeted therapy and may be associated with mechanisms of resistance, and therefore may be an opportunity to study for any potential synergistic activity. So, for example, combinations with EGFR inhibitors or KRAS G12C inhibitors in non-small cell lung cancer would be of interest, as well as combining BRAF inhibitors with TEAD inhibitor in colorectal cancer, or combinations with PI3 kinase inhibitors in head and neck squamous cell cancer. So, in particular, if we look at malignant mesothelioma, we are very excited about some preliminary activity that we've seen in other trials that are also studying TEAD inhibition. And Insilico now has orphan drug designation for our TEAD inhibitor in malignant mesothelioma. So, Sean, dysregulation of the Hippo pathway components clearly plays an important role in tumor progression. Can you tell us how these components fit into the Hippo pathway in human cells?
Sure, Sujata. So in mammals, the core hippo pathway is largely characterized by serine threonine kinases. And these are the mammalian sterile 20 related kinases 1 and 2, or MST1 and MST2, which are actually orthologs of Drosophila hippo. And large tumor suppressor 1 and 2 kinases are LAT-S1 and LAT-S2. These are orthologs of another hippo pathway component in Drosophila called warts. which was named due to the wart-like phenotype it conferred on the flies. So when the HIPPO signaling pathway is active or on, multiple upstream signals activate the cytoskeletal protein NF2, also called myelin, which drives phosphorylation and activation of MST1 and MST2 kinases, which then phosphorylates and activates LAT-S1 and LAT-S2. LAT-S1 and 2 then activate the YAP-TAZ complex, again through phosphorylation. Phosphorylation of YAP-TAZ is an important event as it recruits 1433 proteins that stimulate cytoplasmic retention and proteolytic degradation of that complex, thereby blocking the ability of YAP-TAZ to enter the nucleus. However, when the HIPPO pathway signaling is inactive or off, YAP-TAZ complex is not phosphorylated and instead localizes to the nucleus and forms a complex with the TEAD family of transcription factors. Now, this is especially important in the context of tumor progression, as the TEAD family drive the expression of genes required for cell proliferation, migration, and survival. And for this reason, it's no surprise that many tumors incur mutations in the HIPPO signaling pathway, which result in overactivation of the TEAD family and an increase in cell proliferation and survival that drives tumor growth, and in some cases resistance to anti-tumor therapeutics. So really the TEAD family of transcription factors thereby represent attractive targets from a drug development perspective. So there are 4 TEAD isoforms, each with redundant function, and importantly, all 4 isoforms are expressed in very solid tumor tissues, as shown in this graphic of gene expression across different tumor types. This suggests that approaches to developing drugs against this family may want to target all isoforms in order to achieve full inhibition. In preclinical studies, pan-TEAD inhibitors demonstrate more mesothelia growth, cell growth inhibition compared to TEAD inhibitors targeting single isoforms. And this suggests that pan-TEAD inhibition could be more efficacious than partial TEAD inhibition in a clinical setting. And actually, some early emerging clinical data has been coming out for TEAD inhibitors. Sujata, could you share some of this with us and maybe talk about what this data suggests about the optimal TEAD targeting strategy?
Yeah, that's a good question, Sean. So we know that, for example, we've seen some data that was presented that is testing the TEAD inhibitor that is Vivace's compound in a phase 1 study. That was studying patients with malignant mesothelioma and NF2 mutated tumors. And what they reported, and remember, this is a pan-TEAD compound, that 7 out of 69 patients, mostly malignant mesothelioma patients, had a partial response. They also reported 34 patients as having stable disease. Interestingly, the singleized TEAD1 inhibitor created by Ikena, they have also reported phase 1 study where they have seen as their best response so far 7 out of 7 epithelioid hemangioendothelioma patients as having stable disease, raising the possibility that a pan-TEAD compound may be more efficacious than a singleized TEAD1 compound. So, Sean, as you know, Insilico has a pan-TEAD inhibitor that's entering the clinic. Can you give us some background on the molecule and the preclinical data set?
Sure, Sujata, I'd be happy to do that. So ISM 6331 is a potential best-in-class pan-TEAD non-covalent palmitoylation inhibitor. This novel structure was developed by Insilico Medicine's generative AI-driven drug development platform Chemistry 42. 6331 exhibited potent in vitro and in vivo anti-cancer activities in HIPPO mutant mesothelioma models, one of which is depicted on the current slide. And here you can see our inhibitor shows very good tumor growth inhibition in a HIPPO mutant mesothelioma lung model, with a performance that is markedly superior to the TEAD1 specific inhibitor from Ikena and the pan-TEAD protein-protein interaction inhibitor from Beta Therapeutics. So as you mentioned earlier, Sujata, TEAD inhibition shows great promise for synergy with other targeted therapies such as KRAS inhibitors. And on this slide, we can see some very promising internal data showing strong synergy between our TEAD inhibitor and Amgen's KRAS G12C inhibitor in a lung cancer CDX model. ISM 6331 is orally bioavailable and exhibits excellent DMPK profiles, broad therapeutic index, and good developability. It had good tolerability up to high doses in rodent and dog models based on GLP tox studies and exhibits favorable PK and safety profiles. Importantly, 6331 has a half-life in circulation of around 12 hours, and this should facilitate an optimal dosing schedule in the clinic. So as I mentioned, Sujata, this program is currently going into phase one trials. Can you give us some details on that and how patients potentially interested in participating in the trial could be recruited?
Yes, we're very excited about our phase one trial for our TEAD inhibitor in patients with malignant mesothelioma as well as other solid tumors. We have two parts to this study. One is the dose escalation part, which is primarily to determine safety, and a dose optimization part, which will also give us additional preliminary efficacy information. The dose optimization part is going to be for patients with malignant mesothelioma and other tumors with dysregulation of the HIPPO pathway. This is, primarily to look at all of the information, the safety and the efficacy data to determine our recommended phase two dose. We're very excited to tell you that both in the United States and China that we're actively recruiting patients. We've already dosed our first patient in China. And for those who are listening to this podcast and are interested in participating or referring patients, we have this information here, which is the clinicaltrial.gov site where you can see which sites are open in the United States.
Thanks, Sujata, for that overview. I think you'll agree some very exciting developments going on in the TEAD space, and especially with our own internal program. In addition to our TEAD program, we actually have a strong pipeline of clinical and preclinical candidates across oncology and inflammatory disease. As you can see here, many of these are available for licensing, so we would encourage any interested parties to reach out to us at bd@insilico.com, and we'd be happy to discuss these programs in more detail. So with that, I will thank Sujata for joining and wish everybody a great rest of your day. And we look forward to seeing you on the next podcast.
Thank you, Sean.
Thanks, Sujata.